It is a hard day. So we're back on the clock here, and we will look at the distinction between gram-positive and gram-negative cells. So we're going to use both of these PowerPoints that I mentioned before in passing, and here they are. So you can immediately see a major distinction between these two types. So we're going to look first at the nature of peptidoglycan. Then we're going to look at the gram-positive and the gram-negative cell wall and where they are similar and where they are different. And then understand why that is so impactful. And I was just doing a micro unit for my MCAT, which is the Medical College Admission Test class downstairs. And I because they've done all this before, when, sort of amplify what's going on. So immediately you look at that and you go, they are totally different, and they are. Okay. You have a thick 20 to 40 layers thick of this interesting molecule, polymer is a better way to describe it, peptidoglycan, literally sitting on top of a standard phospholipid bilayer membrane, embedded protein, selectively permeable. We'll start with this. One, it has an interesting chemical that anchors it. It's just sitting there, but you have something to tether it. They're called teichoic acids. The gram-negatives don't have it. 20 to 40 layers, the gram-negatives have 1 or 2 layers. All of this peptidoglycan looks like a mountain or a brick wall, and it's not. It's porous in nature. Okay, so stuff can get through. So you don't need to have passageways for water or small water soluble particles. They can just sort of like drizzle through this immense layer of polymer material. And then you'll have whatever, and the channels will be then here in the standard phospholipid bilayer. So it's relatively simplistic. The interesting thing, again, is this is all a unit, membrane, and that's what they call it, the cell envelope, as you can see here. So they're together. If you distort the membrane, bacteria dies. If you destroy the peptidoglycan that really holds it, it dies. So that's the significant part. With all this peptidoglycan, it makes a wonderfully attractive target for antibiotics because only bacteria have peptidoglycan. So we'll look more closely at that in detail as we analyze and discuss what peptidoglycan is about. We get over here. Doesn't look at all the same. You got a membrane, standard membrane, looks just like this membrane on the inside. We don't even call it the cell membrane. We call it the inner membrane because on the outside we have another membrane, the outer membrane. And the outer membrane, as you can see, has an inner layer that looks just like these two inner layers, this bipolar layer, but the outer layer is totally different. And indeed it is. It's unique. It's not a phospholipid, as you will soon see. It is made of something called a lipopolysaccharide. Just wait and you'll see more about it. So it's not really phosphate as much as it's sugar or polysaccharide based for the outside. You don't see any tethering. It's somehow adherent, and you can see a relatively thin layer of the peptidoglycan. And interestingly enough, in between the outer membrane and the inner membrane and surrounding that peptidoglycan is a unique thing called periplasm. And a lot of interesting stuff goes on in periplasm. For instance, they have what are called basically efflux pumps. So there's pumps utilizing ATP, chemical pumps, that when you put antibiotic close to it, when it gets in here, it pumps it back out, which is pretty neat for a single-celled organism. It's one of the many defense mechanisms they have that make it difficult for antibiotics to work. Moreover, peptidoglycan would be a target. The trouble is, there's a membrane, and not all antibiotics can get through that extra layer. And indeed, we tinker, as you will see in our antibiotics unit, with a variety of base antibiotics. We call them semi-synthetic. The antibiotics, by definition, are naturally occurring. We play with them in the lab to synthesize something that hopefully will get through. For instance, penicillin is naturally occurring. Ampicillin or amoxicillin, which is the orally active form, has been tinkered with in the lab, okay, to be able to penetrate the gram negative. So it creates what we call a broader spectrum. So the spectrum of activity of an antibiotic, if it's narrow, it's either gram positive or gram negative, not likely both, versus broad, which attacks both. So there's a whole, it adds to a lot of nuances that are there. So we don't have the exposed peptidoglycan to attack. We have to somehow get through it if we're going to attack it. And if not, we have to use some other means. And then we're starting to play with fire in the sense that it generates more side effects and potentially more damage in our cells. So that's just the first nugget. In the laboratory, these are a lot harder to stain. When you do the gram stain, the whole idea of the gram stain is trying, we call it a differential stain, 2 different colors so you can differentiate effectively red and blue or red and purple. 2 distinctive colors. And you do it with a couple of things. Well, if we just put blue dye on this, it would be blue. If we put red dye, it would be red. If we put purple dye, it would be purple or green. Okay, this would suck it up. But when we try to use, we first use the blue and then the red, or the red and then the blue, only the last one shows up. The trouble is the gram stain was developed to try to somehow incorporate the first stain in the peptidoglycan. And interesting, in the lab, I'll probably tell you that we still don't really understand how it works. And then we're able to do a maneuver where we can get the second type of cell. So the blue dye, called, it's actually violet, it's called crystal violet, okay, is here. And then we bond it with iodine. And then eventually we use a red dye called safranin here that will incorporate. So these turn blue or purple and these turn red or pink. So there's a lot of that in the chapter. I'm not going to go crazy with it. We should do it in lab. So it's all about the shape, strong structural support. And that's the beauty of where our cells are very sensitive to not enough or too much water. They'll either shrivel or they'll burst. We don't have a big problem with bacteria bursting. They're like a plant. Okay. When you water a plant, it doesn't get sick. It's kind of like, oh, water me. I'm happy. I'm pleasant. Okay. If you don't water the plant, what's going to happen? It's deceased, right? That's one of my wife's favorite yells. You didn't water the plants when I was away for 3 days. In that tone, might I? You killed my plants. So we'll get into why less water is important versus more water. Relative rigidity from peptidoglycan. This is where the illustrations get a little sketchy in this particular one. So with your kind permission or not, we'll move on. So here we are. Same idea, same publisher, McGraw-Hill, gram positive and gram negative. Skip that. Peptidoglycan. Peptidoglycan is a polymer, which chains of identical or similar subunits. You might remember when you had glycogen or cellulose. Except these are nitrogenated carbohydrates. Look at you don't know the chemical. Of course, you can see nitrogen dead. Nitrogen and carbohydrates are good structural material. Our connective tissue uses that for the ground substance. So when you create the matrix of the ground substance and the fibers, nitrogenated carbohydrates, they're structurally sound. Yet there's enough space, so to speak, in there to stuff can go through. They are formed by, and the big N, as you might think, stands for nitrogen. And you don't have to know the name. You don't have to know N-acetyluronic acid or N-acetylglucosamine. Glucosamine should ring a bell. Glucosamine is something that people take as a supplement with arthritis. I take it. So we could call them NAMs and NAGs. So alternating NAM and NAG subunits form a glycan. So sugar with an N, nitrogen. So they sit one on top of the other. But if they're just sitting there, they wouldn't have a great deal of structural integrity. So in order to ensure the integrity, we utilize short chain peptides called tetrapeptides. This means there's 4 amino acids, 3 multi. Okay, and interbridges when we have multiple layers. So we only see the interbridges in gram positives because it allows them to stack. And we don't see it, so the interbridges are like these little guys here. The tetrapeptide groups are in gram negatives. That's not a big deal to us, per se, but understand. So this is what it looks like. So what gives it the structure, the sort of the way I visualize it, it's like this. So you have the bricks, that's the NAMs and the NAMs. Well, if you took a brick and put another brick and another brick on top of it, I mean, it would be substantial, but you could knock it over because there's nothing to hold the bricks in place. You have to use some kind of mortar, some kind of cement. To me, the tetrapeptide groups and the peptide interbridges, they're like mortar. That's kind of, to me, is the first. So it's all about structural integrity. So the interbridges are only in gram positives. You want to remember that, that's fine. That's what you know. So the glycan is the strength, okay, but what holds it together are those peptide groups and peptide interbridges. There's always some in bacteria and gram positives. There's, like I said, average 20 to 40 layers of peptidoglycan, depending on the author you read. And in gram negatives, 1 or 2. So that's why, one way why peptidoglycan is less emphasized in gram negatives as a treatment, things that target peptidoglycan are what we call cell wall antibiotics versus not. So that's the first issue. So it's already complicated. You got to remember all that stuff. You're all looking at me like, WTF did I get myself into with this class? Stands for what the fudge. Just, oh, come on. So relatively thick, teichoic acids are the anchoring material. Okay, gel-like material, there's some, it's kind of like periplasm, but it's really not that distinctive underneath, so it's not like it's sitting directly on top. There's some stuff in between, not a big deal. Which brings us to this disgusting thing. Thin layer, 1 or 2. Don't need the interbridges. Here you see the standard what they call it now, the inner membrane, because there's 2, phospholipid bilayer. Because there's no porosity in a membrane that's inherent, you have to have porin proteins or aquapores. You have to have some type of protein passageway for water to go in and out. So you need them in both membranes, the inner membrane and so to the outer membrane. And just like here, you can begin to see the color. This is a phospholipid. Phosphate heads, lipid tails. Phosphate heads, lipid tails on both sides. Limited amount of peptidoglycan. You know, there's an aspect of lipoproteins that are helping to tether it. It's not teichoic acids. And then you have this outer leaflet, I call it, or outer portion of the bilayer. And it's not a phospholipid, it is a lipopolysaccharide. So it is unique. Unique things are good because they're targets, but unique things are bad because we're unfamiliar with them. And that's the big problem with this. The lipopolysaccharide layer is something that's only in gram-negative bacteria. And the big problem with that is our bodies respond to that as a foreign particle very energetically. So if you have not done the immunology unit in AP2, or for the many of you who are taking AP2 now, you'll be doing that in mid-semester. That's the big problem. We are the lipid A component. We are inherently toxic, or it rather is inherently toxic to us. We respond, but when a gram-negative bacteria gets in an otherwise sterile space, like your abdominal cavity, okay, why would it get there? Ruptured bowel, tumor, ruptured appendix. You would spit out massive amounts of gram-negative bacteria, which are all over the place in your bowel and in the genitourinary tract. And it would explode them literally into your abdominal peritoneal cavity, which is a sterile space, and you would develop something called peritonitis, which is a very, very severe infection that requires intensive care treatment. I mean, it's a ticket to ICU. without even, and we even have a name for it. We call it gram-negative sepsis. When we start talking about when someone is septic, that means the infection has overwhelmed their defenses and they're in big trouble. So sepsis can be from presence of bacteria, presence of toxins, presence of other factors that the body can no longer cope with, if you want to think of it that way. So it's very, very specific. It's a really troubling thing, and it only happens with gram. It doesn't lessen it doesn't mean that gram negatives you can't paint with a broad brush. Gram negatives, because they do this, doesn't make gram negative infections worse than gram positive infections. Okay, because you have some gram negative infections that are comparatively easy to treat, and you have some gram positive infections that are immediately life threatening. Okay. It's just the way it works. So you have to look at them infection by infection, organism by organism in this type of a context. But getting back, this is the component that we respond to. We even give it a name. We call it an endotoxin. Bacteria do their damage by making toxins. So bacteria spit out, it spits out toxins. Doesn't matter what's gram negative or gram positive. Spit them out. They're called exotoxins. It means they're being exocytosed or spit out or released, whatever it is. That outer layer, that lipid A component, is referred to specifically as an endotoxin. We only see endotoxins in gram-negative bacteria. Endo means it's inherently part of its structure. So if a question is phrased, where would you find endotoxins? And let's say, we see it in staph or strep or E. coli. It's only going to be in E. coli. Because E. coli is the only one of those that's gram-negative. So you're going to have to know some of that stuff. And that will help to amplify what you're studying in the lab with what we're studying in lecture. So you're going to have to know the basic groups of the common gram positives and the common gram. And I'll try to emphasize the ones that you'll be dealing with, many of you, on a regular basis, particularly in nursing. Because, I mean, we get a lot of the same infections that predominate. So that's that lipid A. By definition, it doesn't mean that only gram negatives don't make exotoxins. They do. When you hear about that deadly strain of E. coli, that people sometimes they pull food off the shelves when it's contaminated. It's not so much because of the lipid A, it's because it makes particular toxins. Okay. And we're going to get to some of those as well. So the outer membrane of the outside layer is the LPS, or lipopolysaccharide, signals the immune system. So we respond to it immunologically. Okay. If it was just accidentally got in, or a tiny cut, our immune system will deal with it. It's when that appendix bursts, or the bowel ruptures, the tumor, or an ovarian cyst bursts and something bad happens, or an ectopic pregnancy, pick a reason. Okay. Then big trouble. So it's also an endotoxin, large amounts, potentially deadly. It's always recognized. Now, if we go back and take a look at this strand thing here, it's got a core polysaccharide, it's a sugar, and it has what's called an O antigen, and some people call it an O-specific polysaccharide. It's unique to the particular strain of bacteria. So when we're talking about that, there's about 6 really nasty strains of E. coli. The one that you see a lot of stuff being pulled off the shelves and people dying from says E. coli, and you'll see this a number of times. Okay, now we know what it is. O157H7. The O, okay, is not a 0. The O is we are able to immunologically type that O antigen polysaccharide. There's at least 157 of them. So there's a lot. The H has to do with a flagellum, because you're also typing a flagellum. So it's how in the lab, so-and-so's got an E. coli. Let's see what E. coli it is. That way we can tell if there's been contamination in the food supply and they culture it, Is it one that we have to worry about, or is it one that, oh my God, we're in control? And we better have a recall for all that ground meat or whatever was contaminated. Not easy to deal with. Okay, so the outer membrane blocks the passage, including antimicrobes I talked about. Those pores or porins, water, small ions, secretion systems give off those exotoxins that are there. The periplasm, that's what's interesting. Lots of ways that things move in and out in gram negatives go through that periplasm. It's quite extensive, and I don't want to get into it. There's lots of things that occur. And here's and this is other things that why does penicillin work? It doesn't allow the cross-linking of those glycans. You can't get in between them. And we'll go over what the structure is that's there. And lysozyme, if you don't remember it, that's one of the enzymes in a lysosome, but we also have it in tissue fluids to get rid of bacteria that are potentially problematic. And that'll also, and we even use it in the laboratory sometimes for research, because you're trying to destroy the peptidoglycan to analyze the interior. You want to think of it this way. And I'm not going to go into the mechanics of the gram stain, et cetera, et cetera. So that's all. I'm sorry, you got to know all that stuff. Don't look at me like that. I didn't mean to do it. But I think I like you better than the other group. So I'll only make your test. I won't make your test any tougher than theirs. Only if you laugh a lot at my bad jokes.
Okay, we always do that. So.
Yeah. Oh, real harsh. Pardon. Hoarser. Frog in my throat.
Yeah.
Wasn't that funny? My wife, you're using the same stupid jokes you've used for 25 years? Yes. Okay. What? Oh, thanks. Keep. You know what they say? Don't tinkle on my leg and tell me it's raining.
I've never heard anyone say that.
No, they usually say don't pee on my leg and tell me it's raining. Whereas I'm fun. Don't BS a BSer. You've heard that one before. Here, let's see what the Cowan textbook that many of you have unfortunately gotten. I have nothing to do with that. Textbook decisions are above my pay grade. Okay, so it's the same stuff that you see here. Strong and flexible. Sounds nice. Okay, 20 to 80 nanometers. Who cares? There's a couple of variants of teichoic acid in the gram-positive. Cell wall maintenance, enlargement. When you stain, did you talk about basic stains in the lab? You're doing that today?
Start that this afternoon.
Okay. Well, basic is not, they're basic. Basic has to do with acid or basic. So basic stains are positively charged, and they attach to acidic areas of the cell wall, which are negatively charged. Don't tell Dr. Dress I told you.
Will not do that.
Yeah, you're going to see me at the lab. I'm over in the lab next door today. We're experimenting with rabbit muscle.
Wow, with what?
Rabbit
Rabbit what?
muscle.
So cute.
Be very quiet. We're working with rabbit. Elmer Fudd, be very quiet. I'm hunting rabbit. Cartoons? Oh, come on, Grace, you've seen that cartoon. Bugs Bunny?
Yes, I know of it.
Oh, no, you gotta watch that. So, moving on. Okay. It's not the only cell wall in town. The most important one for you, and you will probably stain this in the lab, is mycobacteria. Peptidoglycan, and they're gram-positive, but they're very hard to stain because of this thing called mycolic acid, which is the external shell is not so much of a sugar as it is a fatty acid. And we call it, and the stain we use is called acid fast, not because it's rapid, but it fastens to the mycolic acid. And why is that such a big player? One word: tuberculosis. We work with typically you work in the lab. If they're going to do it, it's a tough stain to do called M. smegmatis, Mycobacterium smegmatis, which is not a pathogen. In other words, it doesn't hurt you. But the two big players in the disease family, and what makes them such a big problem, is this giant capsule, which is very hard to. And as a matter of fact, for tuberculosis, you have to use 2 or 3 antibiotics and rotate them every couple of months because it's highly resistant. Are tuberculosis, and one that you haven't probably seen a case of. I have seen one in my career: leprosy. Very biblical. Very biblical. But I saw a case of leprosy that a soldier on his way back from Vietnam contracted in Africa when he was sent there. I saw him in 1976,
What?
and he was over at the Vets Clinic where I did a program when I was a student in Richmond, Virginia, and it was very, very interesting because his kind of leprosy didn't make all those things fall off that you see biblically and in all the biblical movies, but it was affecting his nerves, and we were doing nerve testing. Interestingly enough, I can remember that case very acutely. So contribute. Pathogenicity means why it's nasty, highly resistant to chemicals and dyes. So you have to go through a whole thing. So archaea, we're really not going to get into because we really don't work with them. They don't have peptidoglycan. They're just oddballs. This is a big player. It doesn't have a quote cell wall. It does have a certain amount of peptidoglycan, but not enough to make it a target. It's very famous, and it's not just confined to pneumonia. But and this is something that affects you folks because it's frequent, and it got its name from military. They called it walking pneumonia because in barracks the soldiers would be there and they would they weren't sick enough, so you'd be able to function. You have a low-grade fever, you have a cough, but it wouldn't stop you from training, so you were able to walk with it. And it takes a long time to get better. You have to do a fairly long course of antibiotics, but you're not at any risk. And about 20% of all pneumonias are this. So mycoplasma pneumonia. We are also seeing mycoplasma, an organism not yet identified completely, playing a role in sexually transmitted diseases. And there's some other forms that I'm not going to get into the old forms. So unusual form. What makes this unusual, when you see an illustration, you may want to have one on the sequence, is it doesn't have that typical rod or spiral or cock's-eye shape. It kind of changes its shape with the environment. So it's really odd. And for that reason, it's resistant to penicillin because it doesn't really have a cell wall, so we can't use cell wall. But Z-Pak works very effectively. So again, the outer membrane, the lipopolysaccharide, signaling molecule, receptors involved with it. It's an endotoxin, the porins we talked about. And that brings us back to really, as we begin to look at the remainder of the internal structure. So, hold on. The chief. So, moving on. So, there's nothing unusual about this particular cytoplasm, whether it's the inner membrane or the cytoplasmic membrane. Same stuff that happens. Understand that in our cells, we have membrane-bound organelles. So energy transformations occur at that membrane. They don't have membrane-bound organelles. They don't have an endoplasmic. So when you're talking about energy reactions, they all occur. Everything is about the cytoplasmic membrane. Even when it's getting ready to divide, the cytoplasmic membrane plays a role as the DNA temporarily attaches to it. It's not a big deal, but you'll find that in biology and molecular structures typically need something as a platform, something to anchor to work. And so they use the membrane a lot. We have a whole elaborate digestive system. They don't. Membrane. Making stuff. Membrane. So regulates transportation of nutrients and waste. By definition, selectively permeable, and the same things apply. Rather than reteaching the wheel, diffusion, osmosis, facilitated diffusion. But I mentioned it before, bacteria, that run, tumble, run, they don't most bacteria are not like us. They're not, there's a limited amount that use aerobic metabolism or true anaerobic metabolism. Most of the time, bacteria don't, and those produce large amounts, relatively speaking, of ATP. Most bacteria ferment. We're going to go into that as we look at sort of the categorization of different types of bacteria. Fermentation makes 2 ATPs compared to 34, 36, 38. So there's not a whole lot of extra energy to expend. That is there. It's useful for us for identification, and that's where it plays a role. So contributes to barriers, the outer membrane. We'll look at the different antimicrobials. Yes, it is harder to deal with in general than a gram-positive. So it makes antibiotic selection more difficult. And you'll see in the laboratory, it makes staining a little more difficult. So it's not that easy. Alcohol-based compounds dissolve lipids in the outer membrane and damage the cell. So that's the neat things about, I'm not a big fan. I don't think alcohol swabs do a whole lot, but in bacteria, certain bacteria are very relatively easy in certain viruses. When we look at viruses, you're going to see that enveloped viruses and bacteria that would classify as, we call them facultative bacteria. We'll get to that. They're easy to grow, but they're relatively easy to kill. Some of them, beg your pardon, fastidious bacteria. And we'll get to those terminologies. So we can cleanse them. So I mean, when you're going to be, so we all are. You know, you're going to give a shot to somebody. You should get that alcohol swab and you wipe it down, start an IV, same deal. Yeah, you're getting rid of stuff on the surface. By and large, you're just cleaning it. It doesn't have to be alcohol. You could use soap and water just as effectively. It really is a little bit overblown. So, and again, you have to have something that can get through that outer membrane to efficiently and effectively treat gram negatives. Extra barrier, genetically difficult. True. On with the cytoplasm. Okay. There's not a lot to it. It's not nearly as, it's roughly the same composition of water as our cytoplasm. And the mixture is, to be fair, relatively complicated. Okay. If I don't do it, and I may, you should, again, take a look at osmosis, diffusion, and facilitated diffusion. It's what I touched on before. Water is an ineffective way to deal with bacteria because they have cell wall. They're really unaffected by large amounts of water, pure water. Removing water is a very efficient way. Historically, the original way to, before they knew bacteria, okay, was to pull water out. We do it to this day. When we cure something, you've heard the term in food science of curing, okay? That's a way that we can even consume raw products. So if you're fond of prosciutto di Parma, okay, which is what's called prosciutto crudo, meaning it's uncooked, but it's consumable. I personally cook it. It's kind of an intricate part of salt and food cooking. But for years, how we dealt with this before we had refrigeration, before we had chemicals, before we had disinfectants, they preserved things in one of two ways: vinegar or salt. When you salted them, you sucked the water out of them. Okay? I say this all the time, a little history for you. The word salary derives from the word salt. The Roman soldiers in the Roman legions were paid with salt. If they didn't get paid, if they weren't worth their salt. A little history for them. So they would salt meats, vegetables. In cooking, when you get, in Italy, when you get the best anchovies, the best capers, if you're into that stuff, okay, they are all packed in salt. You gotta get the salt. That's why anchovies are typically salt. Good Caesar salad needs a little anchovy. Oh, help me. Okay, so the chromosome. The chromosome is this supercoiled aggregate of DNA. Okay, when I say supercoiled, again, it's that twisting of the rubber band thing. The plasmids, definition, useful but non essential. Essential in this context, when something's essential, you require it to survive. So when we talk about essential amino acids, those are the ones we can't get. We have to acquire them through our diet. Plasmids are the same kind of a thing. They're non-essential, the opposite kind of thing. They're useful. Protective traits, drug resistant, toxins, and enzyme production. Ribosomes. Ribosomal RNA, some protein. The typical bacterial ribosome, and I'm sure there's some oddballs, is 70S. We talked about it before, and that's the distinction where antibiotics that address ribosomes or target ribosomes called protein synthesis. So there's two subunits. When they come together, their density changes and it lessens. And the same is true even for the ADS. And they show you the large. And so when they come together, they're a little bit different. You might remember in AP1, they might have talked about how the ribosomes had that strand of messenger RNA and all that assembly of stuff that went on all the proteins. You remember that? Don't worry about it. We're not looking at it. And then other things. Inclusion bodies, food storage, vesicles, things that float to the top of water need air. So the vesicles provide that. Okay. If they're in an adverse environment, those little granules, we use them for identification. Iron oxide with magnetic properties. Some are exotic bacteria are like that. And so there's a lot of things that work with regard. I'm not worried about the micro compartments. Again, we used to think that they didn't have a true infrastructure inside. We know now that they do. Similar to a cytoskeleton that we have in our cells. And yeah, they play a role in the shape. Down the road, this may be something that they'll target. So that's why, and again, that's why we look at these things almost infinitesimally. That's what research is all about. Trying to find something that potentially could be either a source of benefit for us. I mean, to this day, when we talk a lot about antibiotics today, we're constantly going into environments and sampling plants and soil, looking for organisms that might produce new antibiotics since we're running out, new anti-cancer drugs, whatever. We're constantly doing it. That's it's part of what research is about, trying to find things. So we look at these tiny aspects in the hopes that they may produce something. And typically, a research scientist will find something that's sort of like their little niche. Where they like to do stuff like that. Some people are really good. But they study different features. Which brings us to the end. Let's see if we can using it. We got time. Okay. I'll adjust the sound if this is too loud or not too loud. The endospore. The thing about endospores is they're erroneously thought to be a kind of reproduction. Bacteria reproduce by binary fission. They divide in two. So if we have one bacteria now it becomes an endospore, we went from one form to another. It's just a change in form. It's something that a very limited but very significant group of bacteria are able to do to survive in extraordinarily extreme conditions. Effectively, they're probably, if we type them, they've been around for hundreds of thousands of years. Many of them, or they were produced yesterday. Typically, Bacillus and Clostridium, we really don't do a lot with spores Sarcina. We use the term vegetative cells. Vegetative cells derive from plant. When a plant is vegetating, it's growing, it's happy. When a bacteria, which we thought was a plant, a microplant, because it had a cell wall, a vegetative bacteria is growing, happy, producing. It's dormant form, suspended animation, whatever works for you. Okay? That is an endospore. And again, limited. Bacillus in air, that's why we see them in our environment. Clostridium, anaerobic. When they form that spore, once they get into like dead tissue, then all of a sudden you get gangrene and botulism and things like that. We can induce it, but by and large, why would something form a spore in the first place? It forms an endospore, is the proper term, not spore. It forms an endospore in adverse conditions. For you and I, too hot, too cold, whatever. For bacteria, not enough to eat, not enough water. Lack of carbon, lack of nitrogen, lack of water. That's bad news for a bacteria. For me, my internet went out. The game kept cutting out last night. So to kill endospores, because they're living inside, is extreme. And the definition of sterilization is destruction of all living and non-living things, including bacterial endospores. We'll get all to that in a bit. Okay, so here it is. And they have this whole thing, and I'm not going to go through it. Okay, and this is some of the ones we talked about. So I want to show you the animation. That's why I got this. So over here, in the week 1 Blackboard, you have chapter 3 animation zip. If you can't unzip it, give me a holler. This is the MP4, which I use.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into 2 complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by 2 membrane layers. Next, peptidoglycan is laid down between the 2 membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
Okay, just hold that for a while. I'll go over it again with the details. Today. So I'll show you the important. Effectively, what's happening is just transforming. Even though it started to reproduce, because the conditions were adverse, it basically divided asymmetrically. And in the process, formed this endospore. And basically, how do you, I don't know, maybe you have one at home. We have a food dehydrator. So you suck the water out of it, and you can have vegetables and fruits, all that kind of stuff. If you're like a backpacker who hikes, it's a lot easier to do it because generally you can find water. But let me go, I'll walk you through the steps very quickly here as we look without the sound, so to speak. So normally, if under non-adverse conditions, and these for our purposes are both gram-positive, and it's one of the things we look for is to make endospores. Gram-positive rods. That's symmetric, normal. If the conditions are adverse, the DNA divides equally, so it's not a mutation, okay? It's an asymmetric division with scant cytoplasm on one side and an abundant cytoplasm on the other. The mother cell, which is the abundant portion here, envelops, as it continues to grow, what they call the fore or preliminary spore. So now you have a double membrane layer. What gets deposited in there magically? Peptidoglycan at the same time, and this is why it's not reproduction. The other DNA degrades. In other words, just becomes pieces. And when the cell, it just floats around the environment. And you will find them when you do an endospore stain inside the cell and already out in the environment. That's why you're on the bottom of your shoes and in the carpet. So magically, again, this is part of the programming. It makes a chemical called dipicolinic acid. And so once the peptidoglycan, that shell, and it's very, very strong, is the cortex. As the dipicolinic acid forms, okay, inside, it attracts calcium, hardening it, and extrudes water, effectively dehydrating it. And then additionally, as you will see, so there's the water going out and the calcium coming in and blah, blah, blah. And then another protein coat forms to reinforce the cortex and sometimes an additional layer called an exosporion. That's not so terribly important, but to understand that now, when will this change back? As soon as it's got water and food. It's in a friendly environment. Whether it requires air or not depends whether it's Clostridium, no air, or Bacillus, air. And once it gets to that, then it can be effectively restored to its normal vegetative state. So that's everything for today, everybody. And we'll pick it up after that. And that, by and large, concludes most of what we have to talk about the anatomy and the basic physiology of the bacteria for now. Thank you. A lot of